Welcome to the basics of neuroscience. Imagine holding a puppy or hugging a loved one. You feel a wave of warmth and safety. For centuries, we thought this magic came from the heart. But today, we're going to discover that this beautiful feeling actually comes from a highly specific, deeply fascinating chemical symphony in your brain. Let us explore exactly how your brain creates the feeling of affection. Now that we know affection is rooted in the brain, let us look at what it actually is. Affection is not just an abstract emotion. It is a tangible, measurable biological process designed to keep us connected. The neurophysiology of affection studies how our nervous system generates and regulates feelings of social bonding and love. It literally turns the abstract concept of affection into a series of electrical and chemical signals that your brain interprets as pleasure and safety. To understand how these signals work, we need to look at the key players in the brain. At the core of this system are specific brain regions, primarily the hypothalamus, the ventral tegmental area, and the nucleus accumbens. These regions communicate using specialized chemical messengers. The most famous is oxytocin, often called the cuddle hormone, which is released during physical touch to lower stress and promote trust. Vasopressin plays a similar role for long-term bonding, while dopamine provides the feeling of reward. Let us visualize this process with a diagram. On the left, you can see the hypothalamus, which acts as the production center, releasing molecules of oxytocin, shown as blue dots. These molecules travel to the reward center of the brain, specifically the nucleus accumbens, shown on the right. Notice how the oxytocin molecules bind to specific receptors on the surface of the neuron. This exact binding event is what triggers the glowing electrical signal that you consciously experience as warmth, trust, and bonding. Seeing this circuitry in action brings us to an important question. Why did we evolve this way? Understanding the neurophysiology of affection is absolutely critical because it explains the biological basis of human survival. Human babies are born completely helpless, so parents must have a powerful, hardwired drive to protect them. The oxytocin and dopamine pathways ensure parents feel immense reward when caring for their children. Beyond parenting, these neural mechanisms are the glue that holds friendships and entire communities together. Now that we understand the evolutionary purpose, let us look at how we can mathematically predict this feeling. The feeling of affection depends on how many oxytocin molecules successfully bind to receptors. We model this using the receptor occupancy equation. The variable B represents the number of bound receptors, which is the actual strength of the affection signal. The maximum capacity is the total number of receptors available. The ligand concentration is the amount of oxytocin released. Finally, the dissociation constant measures how tightly oxytocin binds. As oxytocin increases, affection increases, but it maxes out once all receptors are filled. Let us see this formula in action with a concrete example. Suppose a person receives a long hug, releasing an oxytocin concentration of 4 nanomolar. Their receptors have a dissociation constant of 2 nanomolar and a maximum capacity of 120 units. We want to find the strength of the affection signal. We plug these values into our equation, 120 times 4 in the numerator and 2 plus 4 in the denominator. This simplifies to 480 divided by 6, giving us exactly 80 bound receptors. Physically, This means a strong, positive affection signal is sent through the brain. This calculation we just did leads us to a fascinating realization about genetics. A person's maximum receptor capacity is determined by their DNA. In neuroscience, researchers study this using voles. Prairie voles mate for life, while meadow voles do not. Interestingly, both species release the exact same amount of oxytocin during mating. The difference lies entirely in their genetics. Prairie voles have a much higher density of oxytocin receptors than meadow voles. Let us calculate how this genetic difference impacts their biological bonding. First, let us calculate the response for the monogamous prairie vole. We are given a high receptor capacity of 200 units. The oxytocin concentration is 8 nanomolar, and the dissociation constant is 2. We plug these into the receptor occupancy equation. The numerator becomes 200 times 8, which is 1600. The denominator is 2 plus 8, which is 10. Dividing these gives us 160 actively bound receptors. This creates a massive, overwhelming reward signal in the brain, promoting lifelong pair bonding. Now we shift our attention to the meadow vole, which has a completely different genetic makeup. We use its much lower receptor capacity of 50 units, while keeping the oxytocin concentration and dissociation constant exactly the same. This isolates the effect of receptor density. Our numerator is now 50 times 8, which is 400. The denominator remains 10. Dividing 400 by 10 gives us only 40 bound receptors. Even though both animals released the exact same amount of the cuddle hormone, the meadow vole's affection signal is 4 times weaker. Comparing these two results perfectly illustrates the power of receptor genetics. The prairie vole experiences a biological affection signal of 160, which is 4 times stronger than the meadow vole's 40. Because the signal is so strong, the prairie vole associates its mate with intense biological reward. This shows how neurophysiology and receptor genetics dictate complex social behaviors like love and attachment. We have now mathematically proven why some animals are biologically wired to mate for life, while others are not. To wrap up our exploration of the neurophysiology of affection, let us review the key takeaways. First, our deepest feelings of love are rooted in precise chemical interactions, driven by hormones like oxytocin and dopamine. Second, the math of receptor binding shows us exactly how these chemicals trigger the electrical signals we experience as trust and bonding. Finally, this system is not just for making us feel good, it is a hardwired biological necessity that ensures our survival and keeps our societies together. You now understand the true science of affection.